Welcome to the third of this series of slideshows that explain the mammalian stress mechanism that was postulated by Hans Selye. This presentation will review the relevant properties of the coagulation cascade enzymes and cofactors that generate thrombin-soluble fibrin and insoluble fibrin. During the era of stress research, relatively little was known about the individual characteristics of coagulation cascade enzymes, including thrombin, tissue factor, and factors 7, 8, 9, and 10, and 13. All these enzymes and substances were assumed to be exclusively involved with blood coagulation, and even now their role in tissue repair remains unrecognized. In particular, the universal requirement for thrombin to energize repair cell activities as well as extracellular enzymes has somehow escaped notice. Though many details remain unclear, fresh information from unrelated research accumulated since stress research was abandoned more than 30 years ago has enabled this simplified diagram of Selye's mechanism that shows how the elegant enzymatic interaction of factors 7, 8, 9, and 10 generates thrombin-soluble fibrin and insoluble fibrin to enable both tissue repair and hemodynamic physiology. Factors 7, 8C, 9, and 10 are all produced by the liver and released continuously into the blood. Factors 9 and 10 have long half-lives so that their blood levels do not fluctuate. Factors 7 and 8C are extremely labile and must be stabilized respectively by tissue factor and von Willebrand factor in order to exert their enzymatic effects. Thus, fluctuating exposure to tissue factor to factor 7 and von Willebrand factor to factor 8C alters the interaction to govern the generation of thrombin, soluble fibrin, and insoluble fibrin. Hemostasis is optional, but tissue repair is essential. Coagulation cascade enzymes generate thrombin to energize tissue maintenance and repair, and clot formation is merely the first step in an orderly sequence of repair activity that restores tissue structures and function. Small amounts of thrombin generated by factor 7 is essential for the activities of factors 8, 9, and 10, which generate additional quantities of thrombin that energize all MSM activities. Tissue factor is a sausage-shaped protein that is tethered to the surface of the cell so that the external portion interacts with blood enzymes. The tethered segment enables cell signaling. Tissue factor initiates factor 7 activity that generates thrombin. All cells appear to have the ability to generate tissue factor, but tissue concentrations of tissue factor reflect hemostasis requirements. Brain, lung, perivascular fibroblasts, peripheral nerves, autonomic ganglia, placenta, and the muscularis mucosa of the esophagus are all rich in tissue factor. The epithelium of skin, mucosa, cardiac muscle, and glomeruli have moderate levels of tissue factor. Little tissue factor is found in skeletal muscle, smooth muscle, excretory ducts, tunica intima, and tunica media. Tissues with high concentrations of tissue factor are targets for stress-related disease manifestations in adult respiratory distress syndrome, multi-organ failure syndrome, eclampsia, and malignancy. All of these illnesses are closely related and could be managed more effectively if their nature were understood. This picture portrays tissue factor binding to factor 7. This stabilizes factor 7 and enables it to generate small amounts of thrombin that energize the activities of factors 8, 9, and 10. Factor 7 thus functions as a trigger for the enzymatic interaction. The complex of tissue factor and factor 7 binds to factor 10. This amplifies thrombin generation and induces factor 10 proliferation. The interaction of tissue factor with factors 7 and 10 is the engine of embryological development and tissue repair, so that defects in any of these three factors are usually lethal. Likewise, anticoagulants such as warfarin and fondoparinux that interfere with this interaction will disrupt embryological development and wound healing as well as clot formation. This explains why they cannot be used during pregnancy. However, treatment with these anticoagulants reduces malignancy risk and enhances the chance of cancer cure. The liver continuously produces factor 8C and releases it into flowing blood. 8C accelerates thrombin generation to convert factor 10 to factor 13. The 8C enzyme is extremely labile and it cannot exert its enzymatic effects unless it is stabilized by von Willebrand factor. Defects in the 8C enzyme cause true hemophilia, which is a severe sex-linked clotting diathesis that mostly afflicts males. The vascular endothelium releases von Willebrand factor into blood in accord with sympathetic nervous system tone and activity. It binds to enzymatic factor 8C to form the chimeric factor 8 molecular complex so that the two molecules circulate together and exert their effects in concert. Von Willebrand factor has no enzymatic properties of its own, but it stabilizes factor 8C and enables factor 8 activity. Defects in von Willebrand factor cause von Willebrand clotting diathesis, which is usually mild, but in some cases it is clinically indistinguishable from true hemophilia. The severity of the clotting diathesis fluctuates in accord with sympathetic tone and activity. Von Willebrand factor also maintains capillary structure, so that von Willebrand factor defects also cause angiodysplasia. Bee stings sometimes trigger sudden complement cascade activity that rapidly attacks and destroys von Willebrand factor, causing anaphylactic shock. This causes hypotension, but cardiac output and tissue perfusion are little affected. It also causes tissue edema that can cause lethal airway obstruction. The treatment of choice is epinephrine, which releases fresh von Willebrand factor from the vascular endothelium. All these phenomena will be discussed in greater detail in later slideshows. Factor 8 is a gigantic chimeric molecule consisting of von Willebrand factor and the vascular endothelium, and from the vascular endothelium, and 8C from the liver that bind together and exert their effects in concert. The rare chimeric nature of factor 8 was unsuspected during the era of stress research. It inspired an extensive review of published literature that enabled the discovery of Selye's mechanism. The unique characteristics of factor 8 functioned as a Rosetta Stone that deciphered stress mechanism function. Both the 8C enzyme and von Willebrand factor are gigantic molecules, and the factor 8 molecular complex is so large that it cannot penetrate the clot of its own making. This limits clot formation to the immediate area of tissue damage. Factor 9 is a cofactor of factor 8. It amplifies factor 8 activity, but it does not affect the other enzymes. Sex-linked defects in factor 9 cause a mild coagulopathy called Christmas disease that has little effect on tissue repair or embryological development. Factor 9 amplifies factor 8 activity, and factor 8 interacts with the complex of factors 7, 8, factor 7, factor 10, and tissue factor to generate factor 13, which converts soluble fibrin to insoluble fibrin. Defects in factors 7, 9, and 13, or excuse me, 8, 9, and 13 are not lethal because they do not disrupt embryological development and tissue repair. Likewise, anticoagulants such as heparin and Plavix that inhibit factor 8 can be used safely during pregnancy because they do not disrupt embryological development. Factor 13 adds crosslinks of fibronectin and plasminogen to soluble fibrin to generate insoluble fibrin monomers that polymerize into strands of insoluble fibrin. Defects of factor 13 are rare and usually mild. It's time for a pause. The next presentation will discuss the vascular endothelium. Thanks for listening.